Introduction. For decades, scientists believed they had a reasonably clear understanding of human evolutionary history. According to the traditional narrative, our species, Homo sapiens, emerged in Africa approximately 200,000 to 300,000 years ago, gradually migrated across the globe, and gradually replaced other hominin species like Neanderthals and Denisovans. However, recent advances in ancient DNA analysis and genomic sequencing have begun to unravel this comfortable narrative, revealing that human evolution was far more complex, interconnected, and surprising than previously imagined. New DNA evidence now suggests that there may be previously unknown human lineages, ghosts, populations, and undiscovered species that played significant roles in shaping modern humanity. These discoveries have profound implications for our understanding of human origins, migration patterns, and genetic heritage. They challenge long-held assumptions about human uniqueness and raise tantalizing questions about lost civilizations and extinct peoples whose existence we only now are beginning to understand through fragments of their genetic code. This article explores the cutting-edge research that is revolutionizing our understanding of human evolution and the hidden lineages that science is only beginning to uncover. The revolution in ancient DNA technology. The ability to extract and sequence DNA from ancient specimens has undergone a remarkable transformation over the past 2 decades. Where researchers once despaired at the prospect of recovering genetic material from bones tens of thousands of years old, modern technology now makes it possible to sequence entire genomes from minimal samples. The breakthrough came with the development of high-throughput sequencing technologies and sophisticated computational methods to distinguish authentic ancient DNA from contamination. Ancient DNA is inherently damaged and fragmented. Over thousands of years, chemical degradation breaks DNA into shorts. Pieces and microbial DNA from soil and handling can obscure the original biological material. Early attempts to sequence ancient DNA often resulted in garbled or inconclusive data. However, innovations in next-generation sequencing, particularly Illumina sequencing and other massively parallel methods, allowed scientists to sequence millions of fragments simultaneously, making it possible to reconstruct genetic information from even highly degraded remains. The turning point came in 2010 when geneticists successfully sequenced the complete genome of a Neanderthal individual using high-throughput sequencing. This was followed by Denisovan genomes, then increasingly ancient Homo sapiens genomes. Each success built upon previous technical achievements, creating a powerful new lens through which to examine human prehistory. Today, researchers can often obtain complete or near-complete ancient genomes from individual specimens, providing unprecedented insight into ancient populations and their relationships to modern humans. The Denisovan discovery. One of the most profound revelations from ancient DNA research has been the discovery and characterization of the Denisovans. Unlike Neanderthals, whose remains had long been recognized as a distinct species, Denisovans were known initially only from genetic fragments. In 2010, scientists analyzed DNA extracted from a finger bone discovered in Denisova Cave in Siberia, Russia, and found it belonged to a previously unknown hominin species. These ancient people, whom researchers named Denisovans, were completely unknown from skeletal remains alone. Their existence was revealed purely through their DNA. The genetic evidence showed that Denisovans were closely related to Neanderthals but constituted a distinct lineage that had diverged hundreds of thousands of years ago. More remarkably, subsequent genetic analysis revealed that Denisovans had interbred with Homo sapiens. Modern populations, particularly in Southeast Asia, Oceania, and East Asia, carry detectable Denisovan genetic material, evidence that these two species coexisted and reproduced together. The Denisovan discovery opened eyes to a crucial insight. Our understanding of human prehistory based on skeletal remains alone was dramatically incomplete. Populations that left no recognizable fossils, or whose fossils had been overlooked or misidentified, could still be detected through genetic analysis. This realization would lead scientists to search specifically for evidence of other unknown lineages, the ghost populations within our DNA. Perhaps the most intriguing evidence for hidden human lineages comes from the detection of ghost genetic segments in modern human DNA, stretches of genetic material that appear to derive from unknown or poorly characterized ancient populations. These segments don't match known Neanderthal or Denisovan DNA, yet they are clearly distinct from the typical human genetic background, suggesting they originated from source populations that are now extinct or undiscovered. In recent years, multiple research teams have identified these anomalous genetic segments. A 2019 study published in Nature described evidence suggesting that modern African populations carry genetic material from an unidentified ancient hominin that contributed to the ancestry of, West African peoples within the past 50,000 years. This unknown population was distinct from both Neanderthals and Denisovans, adding yet another player to the complex tapestry of human evolution. Similarly, researchers have found evidence of what appears to be a previously unknown contributor to the genome of modern Americans. Some of the genetic diversity in contemporary Native American populations cannot be easily explained by known migration events in ancient populations, suggesting an additional source population that remains to be formally identified. These findings have led to hypotheses about undiscovered or underappreciated hominin groups that inhabited the Americas thousands of years ago. The ghost population phenomenon suggests that extinction was the common fate of most hominin species. For every Neanderthal or Denisovan whose DNA we have recovered, there were presumably many others whose remains simply haven't survived, whose fossils haven't been discovered, or whose genetic signatures are only faintly visible in the genomes of their descendants. Detecting these phantom populations requires extremely sensitive genetic analysis and sophisticated statistical methods, but the effort is beginning to reveal the outlines of previously invisible histories. African genetic complexity. While much attention has focused on Neanderthals and Denisovans, equally revolutionary discoveries are emerging. Regarding Africa, the continent where Homo sapiens arose and evolved for most of our species' existence. African genetic diversity is far greater than that of any other continent, yet it remains understudied compared to European and Asian genetics, partly due to historical biases in scientific research. Recent DNA studies of African populations have revealed unsuspected complexity in human origins. Some African populations show evidence of genetic admixture from unknown archaic sources that are distinct from both Neanderthals and Denisovans. The genome of the San people of southern Africa, for instance, contains evidence of ancient genetic contributions that don't match any known ancient hominin population currently represented in genetic databases. This suggests that Africa harbored additional hominin species or populations whose existence we have yet to formally document. Additionally, some researchers have proposed that the origins of Homo sapiens itself may have been more complex than the single origin model suggests. Rather than our species emerging from a single population, genetic evidence hints at the possibility of multiple populations contributing to the emergence of modern humans. These populations may have been geographically separated for extended periods, then come into contact and interbred, with this genetic mixing contributing to the diversity of modern humanity. The Neanderthal-sapiens encounter, revisited. While Neanderthals are no longer unknown lineages, recent DNA evidence has dramatically revised our understanding of the nature and extent of interbreeding between Neanderthals and Homo sapiens. Early studies suggested that modern non-African humans carry approximately 1 to 2% Neanderthal DNA. However, more sophisticated analysis has revealed that the story is considerably more complex. Recent research has shown that interbreeding between Neanderthals and Homo sapiens occurred multiple times, not just once. Different populations of modern humans show different patterns of Neanderthal genetic contribution, suggesting that these encounters happened at different times and in different geographic locations. Some modern populations carry Neanderthal genetic variants that appear to confer advantages in certain environments or immune functions, suggesting that this genetic exchange was not merely incidental, but potentially adaptive. Furthermore, new discoveries have revealed that some Homo sapiens populations may have split off from the main human lineage to interbreed more extensively with Neanderthals before reintegrating with the broader human population. This suggests that human evolutionary history was not a simple march of populations replacing one another, but rather a complex dance of separation, interbreeding, mixing, and movement. Southeast Asia and Oceania. A genetic frontier. One of the most genetically complex regions is Southeast Asia and Oceania, where ancient DNA studies are only now beginning to illuminate the region's deep prehistory. This area harbored some of humanity's earliest long-distance maritime voyages and the peopling of remote islands. Yet until recently, we knew almost nothing about the ancient peoples who undertook these remarkable journeys. Recent genome sequences from early inhabitants of Southeast Asia, the Philippines, and Oceania have revealed previously unsuspected genetic complexity. Some of these ancient individuals show a mosaic of genetic contributions from multiple archaic sources. A 2021 study described a previously unknown hominin lineage detected in the genomes of some Philippine populations, a ghost lineage that appears to have interbred with anatomically modern humans, but whose skeletal remains have never been found. The Denisovan genetic contribution is particularly notable in Oceania. Some Polynesian and Melanesian populations carry substantial Denisovan genetic material, approximately 4 to 6% of their genomes, far more than most other populations. This suggests that Denisovans survived in Southeast Asia and Oceania for far longer than in other regions, and that they played a significant role in the ancestry of Pacific Islander populations. As researchers discover more ancient DNA from these regions, the picture becomes increasingly complex, revealing multiple waves of migration, local interbreeding events, and the contribution of unknown populations to the ancestry of modern inhabitants. The American enigma, perhaps nowhere, is the evidence for unknown lineages more puzzling than in the Americas. The peopling of North and South America represents one of humanity's greatest achievements, the colonization of vast, previously uninhabited continents. Yet the genetic and archaeological evidence for how this occurred remains surprisingly mysterious. Traditional models proposed a single migration event via a land bridge, Beringia, connecting Asia to North America approximately 13,000 to 15,000 years ago. However, genetic evidence has become increasingly inconsistent with this simple model. Modern Native Americans show genetic signatures that are difficult to explain through a single founding population. Some researchers have detected genetic material in Native American populations that appears to derive from ancient populations that share closer affinity with European or East Asian populations than would be expected from a single migration event. A controversial hypothesis proposes that some of the earliest Americans may have had ancestry from Solutreans or other Ice Age European populations, or from populations with East Asian connections that diverged from the ancestors of modern East Asians very early in human expansion. While this hypothesis remains disputed and requires further evidence, the underlying genetic data suggesting multiple or more complex population sources is becoming harder to dismiss. Additionally, some research has detected what appears to be ancient genetic material from unknown sources in Native American populations, genetic variants, and segments that don't match any known ancient or modern population in currently available databases. These orphan genetic segments might represent contributions from extinct populations that inhabited the Americas and whose descendants left no other trace in the genomic record. Homo floresiensis and other marginal cases. The discovery of Homo floresiensis, a diminutive hominin species standing only about 3.5 feet tall and known from fossils found on the Indonesian island of Flores, demonstrates that our understanding of human relatives remains incomplete. Despite their skeletal remains being well documented, genetic material from H. floresiensis has proven extremely difficult to recover due to the tropical climate of their island habitat, which accelerates DNA degradation. The absence of genetic data from H. floresiensis is frustrating because this species raises profound questions about human diversity and the variety of ways that hominins solve the problems of survival and reproduction. If H. floresiensis were as distantly related to other hominins as its skeleton suggests, yet coexisted with modern humans for thousands of years, this would indicate that human evolution was even more diverse and complex than currently recognized. Relationships to other hominins, and whether they interbred with Homo sapiens. Methodological advances enabling discovery. The acceleration of discoveries regarding hidden lineages has been driven by methodological innovations beyond simply extracting and sequencing ancient DNA. Sophisticated statistical approaches now allow researchers to detect very small amounts of genetic contribution from ancient populations, contributions that would have been invisible using older techniques. One particularly powerful approach is called admixture mapping, or chromosomal painting, which involves identifying stretches of DNA in modern individuals that appear to derive from ancient populations. By scanning the genomes of diverse modern populations and identifying segments that don't fit the expected pattern, researchers can infer the existence of hidden genetic contributions and sometimes even estimate when the interbreeding events occurred. Another innovation involves using modern human genetic data to infer the properties of ancient populations. By studying patterns of genetic variation in living humans and using computational methods to trace these patterns backward through evolutionary time, researchers can sometimes detect evidence of ancient populations that are not represented by skeletal fossils or ancient DNA. This approach has sometimes led to surprising discoveries about the deep structure of human genetic diversity. Additionally, researchers have developed methods to extract DNA from unexpected sources. Sedimentary DNA, genetic material preserved in soil and sediment layers, has proven to be a rich source of ancient genetic information. By analyzing DNA recovered from archaeological sites, scientists can sometimes detect evidence of species that inhabited an area based purely on traced genetic material without requiring skeletal remains. Implications for understanding human nature. The revelation of hidden human lineages carries profound implications for how we understand ourselves. First, it demonstrates that our species did not emerge fully formed, but was shaped by encounters with other human-like populations. We are, quite literally, hybrids, products of our species' encounters and interbreeding with other lineages. This genetic inheritance is not incidental, but constitutes a real part of who we are, sometimes conferring adaptive advantages in particular environments. Second, these discoveries underscore the importance of genetic diversity and the fact that human populations have always been fluid and interconnected. The notion of pure or isolated human populations is revealed as a fantasy. Even in prehistory, thousands of years ago, human populations encountered one another, exchanged genes, and influenced one another's evolution. Modern concepts of human populations as fixed or biologically distinct are fundamentally at odds with our evolutionary history. Third, the evidence for multiple hidden lineages reminds us of the vast extent of our ignorance regarding human prehistory. For every species we have documented through skeletal remains or genetic data, there may have been others that left no trace, or whose remains remain undiscovered, or whose existence we can only infer indirectly through genetic fragments in modern populations. This should encourage both humility about our current knowledge and excitement about future discoveries, future directions, and unresolved questions. The field of ancient genomics remains in its infancy. Many questions that would have been unanswerable even 5 years ago can now be addressed, but vast domains of mystery remain. Key future directions include geographic expansion. Most ancient DNA research has focused on Eurasia and Africa, with less coverage of the Americas, Southeast Asia, and Oceania. Expanding ancient DNA sampling to these regions will likely reveal additional hidden lineages and complexity in human prehistory. More complete genomes. While researchers can often recover substantial portions of ancient genomes, truly complete, high-coverage sequences from ancient individuals remain rare. As coverage improves, it becomes possible to detect smaller contributions from ancient populations and to resolve finer details of population history. Environmental and phenotypic DNA. Most current research focuses on tracing population history and admixture, but ancient DNA also carries information about the traits and adaptations of ancient peoples, what they look like, how they were adapted to, their environments, and how they changed over time. Decoding this information will provide richer understanding of prehistoric human populations. Integration with other data. Ancient DNA must be integrated with archaeological evidence, skeletal morphology, linguistic data, and paleo-environmental information to build comprehensive pictures of human prehistory. Interdisciplinary approaches will likely yield richer and more accurate understandings than any single data type can provide. Ethical and social implications. As more genetic information about ancient peoples is recovered and analyzed, questions of cultural affiliation, indigenous rights, and the appropriate use of genetic data from human remains become increasingly important. Moving forward, this research must be conducted with sensitivity to indigenous communities and consideration of the broader social implications of genetic discoveries.